Good afternoon. My name is Thomas Farr-Porce, CEO of Boston-based Thrive Bioscience, and started Thrive to fill the need for analytics and automation of cell culture, as well as stem, tissue, and pathogen culture. Thrive's instruments, for the first time, provide sequencing quantities, but for live cell biology. We've started selling a benchtop imager and an automated 50-plate imager, each of which captures thousands of images in minutes, analyzes, and builds a centralized database. And a result of this data are custom research problems. For example, the University of Texas Medical Branch in Galveston, for the first time, is now able to study and measure the effects of viruses on cells in as little as 2 days, instead of the current 5 to 7 days. This capability will be one of several separate software application packages, along with imaging organoids, monitoring single cells. We've raised $25 million to date, and we've just started raising a $12 Series B for sales and to complete packages. Thank you.
What is the valuation, sorry?
So we haven't set the valuation. The post-money valuation on the Series A was approximately 45 million US dollars.
I'm sorry, Tom, could you just sort of refresh my memory? Is it just software, or it's software and hardware combined?
Sure. So it is software and hardware combined with a very high software component. So we have a family of instruments, benchtop, a 50-plate imager combined with an incubator, and then we're working on a full automation. And it's really because of the integration, we're able to emit 1,000 images. We take user selectable between 5 and 50 focal planes. Each image is 5 megapixels. And then we take that data, send it by network to an analysis workstation where we stitch them, create metrics, make it easy to mine the data, and you're able to look at it over time. So the plates are taken out and put back in. Our image registration is so good that you get back to within a cell distance of a cell or a colony. So you're able to see the progression of viruses on cells or drugs on cells.
And I guess because you said you can take the images in minutes and then there's some sort of data processing component, can you theoretically assess the cells in real, would you?
So close to real time. So we're computing all kinds of metrics on the growth rates, the size, the shapes, the depth. Some of our customers have used it for drug response, and we're producing metrics that that show them things they haven't seen before, and as they're viewing the cells on a tablet in the lab, and then on an analysis workstation in their office, or it's web browser Excel. So you can be halfway around the world and looking at your cells as well as your database of cells.
Right. And I guess my next logical question, I suppose, is given the fact that you can collect all of this data about the cell, have you explored applying machine learning algorithms now to sort of add information to the researchers, right? To sort of consolidate that information and give them meaningful insights.
Every one of our customers says that they have had have solved research problems they were not able to before because of the amount of data. We've enabled their use of AI, but we also have a package that will be one of our software modules that allows them to train the system to look for things that they believe are meaningful, or to find correlations using neural networks and other statistical methodologies to find correlations, say, between shape of a cell and whether or not the cell isn't. My own background, the first 3 companies I started were actually AI companies. One of them was a multi-billion-dollar exit.
Interesting. Okay, thanks.
We're very much at the intersection of imaging and technology and analysis, data mining, machine learning.
Tom, when you will break even now with the current financing round?
So we expect just a few $1 million of sales this year. We just hired a commercial team, but we expect in 2 years to be profitable.
How many clients or customers do you need for reaching this break even?
So we have a mix of high margin software and high margin, well, actually, and high margin instruments. And in order to get to break even, we need to sell approximately 200 of these instruments over approximately 2.5 years. And there are thousands, a very large market. Cell culture is performed throughout the world and is the base of much of medical research. And we're applicable to tissue culture, stem cell culture, embryos for in vitro fertilization.
Okay, thank you.
You asked everyone else about their IP. Hoping. We have, because these instruments really have not been developed before, there's just been so little innovation in cell culture. The companies I started, the Cytic, where we automated cytology labs, smears, was very similar. There were just no tools. And so here, these instruments and tools just have not been developed. So the IP field's really wide open. And we've filed 65 patent applications in 25 families, and it covers AI, blockchain, IoT, collecting all the information within a cell, algorithms. We think we have the world's best algorithms for focusing. We don't focus on debris. We have IP on aggregating and comparing data across microscopes, instruments, other lab instruments. So we think we have a strong position.
Tom, and given your experience, how you envision an exit?
So this is an area where is of great interest to 10, 15 companies. Thermo, Sartorius, Dedham, Danaher, any of the imaging companies, any of the instrument companies. One of the big wins here is that right now we're spending billions every year on instruments and assays, and we're using that on cells that we just out of the air that we know almost nothing about. They're often mislabeled. We don't know the population. There's tremendous variation, and so we actually make other instruments work much better and with much less variability. So we fit well as an acquisition. Public multiples. I've run public companies before. Public multiples for tools really excellent. So either way, but I think we're really quite valuable to the instrument or imaging companies. We're really in some ways a 4th generation or whatever generation microscope, but we also have a family of products. Our next product, which is in alpha test, walk-away automation, it's basically fluidics and mechanics driven by our cells. It all is down really imaging these cells, making decisions about them, analyzing them, and then driving the automation.
So would that be more of a closed system then? And could you GMPize that then?
Yes. So our chairman, Guy Broadbent, was president of Thermo Fisher, and then was recruited by Kleiner Perkins into Accelerex, which was bought by GE. It's one of their best acquisitions, and it's cell culture on the bioprocessing side. And this is a tremendous opportunity for us. Currently, we're in the research market. We're getting pulled into the bioprocessing. Our customers are having extraordinary results, increasing yields and measuring quality. By having all of this data, they can see wherever there's outlier. One of the software packages I didn't mention is everyone in the industry, in the field, growing stem cells tries to pick monoclonal colonies, but nobody knows whether a colony is monoclonal. There's no way that you go back in time, but you can with ours. You can go back and look at time series data of any colony, pick on your tablet or on a computer. And we're about to come out with a package that just automatically tells you all colonies that are monoclonal. So you pick the ones that are monoclonal. So we're having tremendous impacts on quality and yield and cost, because you can compare to reagents in the research world. We're bringing some of the kind of rigor of bioprocessing and GMP to research, but our goal is to move exactly as you say into
bioprocess. Yeah,
it's a different market. It's longer sales cycles, but very sticky, and you have to be validated. So we need a lot of data from research customers.
Yeah, definitely very sticky though, right? Because once you get baked into the process, then they have to use you, right?
Kind of an annuity.